Imagine finding out that, the chronic pain and incontinence affecting millions of postmenopausal women isn't just a simple matter of aging tissue.
Right, like it's just normal wear and tear.
Exactly. But what if it's actually a microscopic cellular strike? Like where the workers inside your body are literally passing out memos to stop rebuilding the tissue.
It's a crazy concept. I mean, for decades, we've viewed our anatomy as this static, fixed architecture.
Yeah.
But under the surface, your tissues are this bustling 24/7 construction site. And today, we're looking at what happens when the communication at that site just totally breaks down.
Specifically in the pelvic floor, right. And more importantly, how cutting-edge science has figured out a way to rewrite those biological memos. So the mission of our deep dive today is to really explore the specific molecular mechanisms of how these things called mesenchymal stem cell-derived exosomes,
Which are basically microscopic cellular text messages.
right, how they actually upregulate elastin and collagen gene expression to rejuvenate postmenopausal vaginal fibroblasts. We're looking at a pretty hefty stack of sources today.
Yeah, we've got everything from molecular biology journals like Pelvic Perineology and PLOS1 all the way up to early clinical trial data.
And the context here, it impacts millions of individuals. When you go through menopause, there is a systemic drop in estrogen.
It's a huge drop, yeah. And that hormonal shift triggers severe structural changes. So we see things like vaginal atrophy, pelvic organ prolapse, or POP, and stress urinary incontinence, which is SUI.
And historically, if you went to a doctor with those conditions, the focus was mostly on the macro level.
Right, treating the big visible symptoms.
Exactly. The options were often systemic hormone replacement therapy, which, floods your entire body with hormones, or invasive reconstructive surgeries involving transvaginal meshes.
And those meshes have famously had a lot of severe complications over the years.
Yeah, a lot of controversy there. But the sources today reveal a completely different cell-free regenerative approach. Okay, let's unpack this. To understand how this new therapy actually fixes the problem, we first have to zoom way, way in to look at what is physically breaking down at the cellular level.
Yeah, so in the pelvic floor and the vaginal wall, the primary builders of the tissue are these cells called vaginal fibroblasts.
Okay.
And their sole job, their entire purpose, is to construct and maintain the extracellular matrix, or the ECM.
I always picture the extracellular matrix as like the steel framework of a skyscraper.
That's a great way to look at it.
Yeah, so the fibroblasts are the construction workers on the site. They're laying down the steel beams, which is the collagen, and that provides the firmness and the tensile strength.
Right, but a completely rigid steel building would just snap under pressure.
Exactly. So they also have to lay down flexible joints. And that's the elastin, which allows your tissue to stretch and recoil naturally.
But in conditions like POP and SUI, that architectural blueprint basically goes awry. The research shows an abnormal distribution and a seriously decreased quality and quantity of those fibroblast workers.
So you have fewer workers, and the ones that are there are exhausted.
Yeah. And the problem is compounded by a specific class of enzymes called matrix metalloproteinases, or MMPs, specifically MMP1, MMP2, and MMP9.
The wrecking crew.
Exactly. These enzymes are naturally designed to act as a cleanup crew,
Yeah.
Clearing away old, damaged tissue so new tissue can be built. But postmenopausal estrogen withdrawal somehow uncouples their regulatory mechanism.
It just sends them into hyperdrive.
Complete.
So the MMPs become this overzealous demolition crew that just will not stop tearing down the framework. You've got fewer tired builders on the scaffolding, while a massive wrecking crew destroys the collagen and elastin fibers way faster than they can ever be replaced.
And it gets even more hostile. If you look at the study published in PLOS1, the researchers found that in patients with stress urinary incontinence, their own vaginal fibroblasts actually begin secreting bad, small extracellular vesicles.
Wait, bad vesicles?
Yeah, these vesicles travel to neighboring cells and actively inhibit collagen synthesis and cellular migration. The local environment essentially turns against itself.
Wow, so the exhausted workers aren't just slowing down. They're actively printing out memos, handing them to the other workers, and telling everyone on the site to stop building.
That's exactly it. And this
Exactly. We have to somehow change the instructions they're receiving. And I think that brings us to the core solution highlighted across all our sources today, which is mesenchymal stem cell exosomes.
Right. Mesenchymal stem cells, or MSCs, are adult stem cells known for their intense regenerative properties.
And where do they come from?
Well, the literature shows they can be safely harvested from a few different tissues. Adipose, or fat tissue, is a very common one. They can also be derived from the umbilical cord, and remarkably, even from menstrual blood.
Really? Menstrual blood?
Yeah, it's actually emerging as a highly potent source for these specific pelvic therapies.
Okay, but wait, let me push back on this for a second. If the local tissue is failing and the native cells are totally exhausted, shouldn't we just be implanting fresh, healthy, living stem cells directly into the tissue to do the heavy lifting?
That's what people usually think, yeah.
Right, so why are the sources so focused on their exosomes? I mean, if an exosome is just a tiny bubble, how can it possibly do the work of a whole living stem cell?
Well, what's fascinating here is that the healing power doesn't necessarily come from the physical presence of the stem cell itself. It comes from the information it secretes.
Oh, I see.
Yeah, exosomes are not living cells. They're extracellular vesicles. You can think of them as nanoscale cargo ships. A stem cell packs these tiny bubbles with a highly curated payload of lipids, proteins, messenger RNA, and microRNAs, and just sends them out into the tissue.
So it's biological mail.
Exactly. And by using only the mail, rather than the cell that sent it, researchers achieve what's called a cell-free therapy.
Which has a lot of advantages, I'd imagine.
Massive advantages. Living stem cells are bulky, they can be unpredictable, and if they come from a donor, they carry cellular markers that flag them as foreign invaders.
Which triggers an immune response.
Right. But exosomes lack those specific immune-triggering markers. They have virtually zero risk of immune rejection, very low toxicity, and because they're nanoscale, they can penetrate deep into dense, damaged tissues way more effectively than whole cells.
So they just slip past the immune system's security guards, find those exhausted local fibroblasts, and deliver a completely new set of instructions.
Yep.
So how does this microscopic biological mail actually reprogram a postmenopausal vaginal fibroblast?
Well, when an MSC exosome enters the tissue, the first thing it does is trigger a massive upregulation of a specific gene called COL1A1.
COL1A1.
And that's
Yeah.
the gene directly responsible for manufacturing type I collagen, which, going back to your analogy, is the heavy-duty structural steel of our tissue scaffolding.
Right. And the mechanism driving that upregulation relies heavily on microRNAs, or miRNAs, right?
Yes. The detailed study we have from pelvic perineology points out that in patients with pelvic organ prolapse, specific microRNAs, like one called miR1295P, are found at abnormally high levels.
And those are the bad memos.
Right. These elevated microRNAs actively suppress tissue repair. So the MSC exosomes counter this by delivering beneficial microRNAs that alter the cell's signaling pathways. And a primary target here is a cytokine known as TGF-beta1, or a transforming growth factor beta-1.
Okay, so if we think of TGF-beta1 like the site foreman at our construction site, its job is to shout the orders to ramp up collagen production.
Exactly.
So the beneficial microRNAs from the exosome basically hand that foreman a megaphone, amplifying the signal so the tired fibroblasts finally wake up and get back to work.
That's a perfect way to put it. But, generating new collagen is completely futile if that hyperactive demolition crew, the MMP enzymes, is still on the site tearing it all down.
And here's where it gets really interesting. The exosomes don't just hire more builders by upregulating collagen genes, right? They actively fire the demolition crew simultaneously.
Yes, they do.
Because the sources show that when these fibroblasts absorb adipose-derived MSC exosomes, it triggers a massive spike in the production of TIMP1 and TIMP3.
Right, those are tissue inhibitors of metalloproteinase.
Basically, they are the direct molecular brakes for those destructive MMP1 and MMP2 enzymes.
Exactly. The exosome drops off a piece of genetic mail that tells the cell, hey, start manufacturing the specific inhibitors that will shut down the wrecking crew.
It's a dual action mechanism.
Right. You increase the synthesis of the scaffolding while actively halting its degradation. And this dual action is really the essence of restoring cellular homeostasis.
And there's a really critical nuance here regarding the type of scaffolding being built, right?
Yeah, because the body requires a very specific balanced ratio of collagen type 1 to collagen type 3 to maintain a healthy pelvic floor.
Otherwise, things get messed up.
Exactly. Modulating those microRNAs ensures the fibroblasts restore that optimal ratio. If you only stimulate unorganized collagen production, the tissue just becomes fibrotic,
It just turns into stiff scar tissue.
which is completely useless for an organ that needs to expand and contract. So this necessitates elastin. The research heavily emphasizes that these exosomes don't just stop at collagen, they also upregulate elastin genes and a really crucial enzyme called LOXL1, or lysyl oxidase like 1.
Ah, yes. I was really hoping we'd get to LOXL1 today. Because building flexible joints isn't as simple as just dumping elastin proteins into the tissue, is it?
Not at all.
The elastin fibers have to be properly connected to actually function. I mean, without LOXL1, producing elastin is like dumping a massive pile of bungee cords onto a floor. They're stretchy, sure, but they don't provide any structural support until you meticulously hook them all together into a cohesive, load-bearing net.
Yeah, and LOXL1 is that molecular hook. It facilitates the cross-linking of those elastin fibers. And just to understand how vital this is, researchers have actually engineered mice to completely lack the LOXL1 gene.
And what happened?
Those mice invariably develop massive pelvic floor disorders,
Yeah.
simply because their tissues cannot handle everyday mechanical stress.
Wow.
So by upregulating both the raw elastin and the LOXL1 required to cross-link it, the exosomes restore the dynamic biomechanical function of the vaginal wall. They aren't just patching a hole, they're rebuilding the functional net.
Okay, so the scaffolding is secure, the fibroblasts are laying down cross-linked elastin, they're pumping out the right ratio of collagen, and the rogue demolition enzymes have been completely neutralized. But a scaffold is just a physical structure. For any tissue to survive, it requires a healthy local ecosystem. I mean, if the environment around the scaffold is starved of oxygen or highly inflamed, those newly active fibroblasts will just burn out again.
Yeah, they wouldn't last long. And this brings us to the secondary effects of the exosome payload, starting with angiogenesis, which is the creation of new blood vessels.
Okay, so how do they do that?
Well, the data shows that these nanoscale cargo ships deliver potent pro-angiogenic factors, most notably VEGF, or vascular endothelial growth factor.
VEGF.
Got it.
Right.
And VEGF acts as a chemical beacon to the endothelial cells lining nearby blood vessels, signaling them to branch out and form new capillary networks.
And in the clinical studies, researchers actually measure this new plumbing system by looking for a marker called CD31. So more blood vessels mean more oxygen, and nutrients are delivered directly to our newly revitalized construction workers.
Exactly. But it goes beyond just blood flow. The patent data in our sources outlines a pretty fascinating solution to one of the most frustrating symptoms of menopause, which is the loss of vaginal pH and lubrication.
Yeah, because menopause typically destroys the naturally acidic environment of the vagina, which causes extreme dryness, painful intercourse, and a really high susceptibility to chronic infections.
And the patent data reviews that when exosomes are introduced to the vaginal lining, they trigger an immediate increase in intracellular molecules known as TAMP and cGMP.
Right. And to put that in non-jargon terms, you can think of CAM&P as like a cellular fire alarm or an internal metabolic thermostat. When those levels spike, it completely changes how the cell processes energy.
The technical term for that is an increase in glycolytic flux. The thermostat tells the cells to start breaking down glucose at a much higher rate.
And the byproduct of that accelerated glucose breakdown is lactic acid.
Exactly.
And that surge in lactic acid is precisely what restores the protective acidic pH shield that menopause stripped away.
Furthermore, this metabolic shift stimulates the production of transidate.
Which is?
Transidate is the fluid that naturally lubricates the vaginal walls from the inside out. So by turning up that metabolic thermostat, the exosomes dramatically decrease dryness and without the need for topical synthetic lubricants.
That's incredible. But, delivering these nanoscale bubbles into the vaginal canal presents a major physical challenge.
It really does.
Because the mucosal lining of the vagina is designed to constantly slough off and clear things out. If you just spray a liquid full of exosomes in there, gravity and natural fluid flow will wash them away long before they can penetrate the tissue and reprogram the fibroblasts.
Yeah, they'd be gone in minutes.
So how do researchers actually keep the exosomes on site long enough to do their job?
Well, that delivery challenge is solved by embedding the exosomes into a hydrogel. A hydrogel is a water-swollen, cross-linked polymer network. It essentially acts as a bioadhesive scaffold.
It's like a sticky microscopic sponge.
Perfect description, yeah. When applied to the vaginal wall, the hydrogel adheres to the tissue and creates a slow-release matrix. So instead of washing away, the exosomes slowly diffuse out of the gel over time. This ensures a sustained delivery of that genetic payload deep into the fibroblast layer.
That makes total sense.
Yep.
Let's look at how this plays out in the real world, though, because we've been talking heavily about microscopic mechanisms. How does this molecular reprogramming actually translate into physical relief for a patient?
Well, the University of Texas Southwestern Medical Center ran a really compelling study on this.
What did they do?
They created a menopause model using rats. By removing the rat's ovaries, they induced a sudden estrogen drop that perfectly mimics the onset of severe vaginal atrophy. Then they applied umbilical cord MSC exosomes locally using that exact hydrogel delivery system.
End result.
The tissue analysis following the treatment was striking. They observed a significantly thicker, more robust vaginal epithelium. They also measured a massive increase in a protein marker called Ki-67.
And just for context, Ki-67 is basically a molecular snapshot that proves cells are actively dividing and regenerating rather than just sitting dormant, right?
Exactly. Finding elevated Ki-67 means the tissue is actively rebuilding itself. And similar outcomes were documented in studies involving macaque monkeys.
Which is important because macaques are vital for this research. Their reproductive tracts and hormonal cycles are structurally much closer to humans than rodent models.
Right. And injecting MSC exosomes into the macaques significantly increased microvascular density, along with the dense networks of collagen and elastin we just discussed.
And we're even seeing this transition into early human clinical data now. The sources detail a study utilizing something called the Cephagy-YN device.
Yet the Cephagy-1 approach involves using mechanically fragmented adipose tissue.
Okay, so fat tissue.
Right. But instead of using harsh enzymes to break down the patient's fat to extract stem cells, they mechanically process the fat. This preserves the delicate microenvironment, which is naturally rich in these regenerative exosomes and microRNAs.
So they keep the good stuff intact. And when this mechanically fragmented tissue was injected into postmenopausal women, the researchers tracked them over a 5-month follow-up period.
And the results were really promising.
Yeah, they noted a significant attenuation of vaginal atrophy symptoms and massively improved vulvar trophism, which essentially means the overall health, thickness, and nourishment of the tissue was restored. The patients experienced highly effective, non-hormonal relief from pain, dryness, and incontinence, precisely because the therapy rebuilt the architectural integrity of the tissue from the inside out.
If we connect this to the bigger picture, it represents a fundamental evolution in how we view healing. Instead of fighting the body's natural breakdown by inserting synthetic meshes or flooding the system with systemic hormones, we are learning to supply the exact biological blueprints needed to trigger natural, highly localized regeneration.
It's amazing. Let's do a quick recap to pull all of this molecular biology together for you. We started by zooming in on a microscopic construction site in crisis.
Right, where a systemic drop in estrogen causes tired vaginal fibroblasts to stop building the pelvic floor scaffolding.
While those rogue MMP enzymes act as a demolition crew, tearing down the existing framework and causing conditions like POP and SUI, then we introduced mesenchymal stem cell-derived exosomes, not as living cells, but as nanoscale messengers packed with genetic instructions.
Delivering the mail.
Yes, delivering the mail. Using a hydrogel to hold them in place, these tiny cargo ships slip into the tissue without triggering an immune response. They rewrite the biological memos, upregulating the COL1A1 gene to build new collagen, upregulating LOXL1 to cross-link flexible elastin, and triggering TIMPs to finally fire the demolition crew.
And through factors like VGF and CAMP, they build new blood vessels, restore the acidic pH, and bring back natural lubrication.
It really is the ultimate form of localized, cell-free repair. We are treating the microenvironment to solve the macro problem.
It completely flips the script on aging and tissue degeneration.
It really does. And that leaves us with a final thought to ponder as we wrap up today's deep dive. If science can now isolate these exact cellular text messages, these specific exosomes to perfectly reprogram aging vaginal tissue, could we eventually map and sequence the unique exosome language of every single organ in the human body?
That's the dream, isn't it?
Imagine a future where structural breakdown isn't treated by trying to alter the physical cells themselves, but simply by changing the mail they receive, where we send the right message to fix the scaffolding long before the building ever truly falls apart.